Greetings everyone. Today we will discuss about monoclonal antibody and its production by hybridoma technology. Now, what is monoclonal antibody? Monoclonal antibodies are those antibodies which specifically recognize an antigenic determinant. So now these antibodies, as I told, is very much specific. It can recognize a small chain of 5 to 20 amino acid sequence. So now, when an antigen enter into our body, antibodies are produced against it by B lymphocyte. Antigen, when it enter into our body, inside the body, the B lymphocytes produce antibodies denoted as AB, antibodies. So now these antigen, they generally have multiple antigenic site. Antibodies are produced against each of these sites. For this one, some B lymphocytes are, these are indicative of, for each one B lymphocyte will be involved, for each one. This B lymphocyte will specifically produce antibody against this one, this antigenic site, which is 5 to 10 amino acid sequence, 5 to 20 amino acid sequence. So now this one, though all will produce antibodies. So if we indicate them as 1, 2, 3, 4, all these antibodies will react with this antigen. All these antibodies will react with this antigen. So they are not that much specific. They are specific but not specific as the individual ones. This antibody will always react with this one only, not with other 3 antigenic site. So now if we cultivate this B lymphocyte outside our body, then what will happen? These cells will produce antibody which are specific to this one only. That is, the clones of these B cells will produce antibody which is specific to these cells. These antibody, if cultivated outside our body, is called monoclonal antibody, indicated as so there is a problem in cultivating these B cells outside our body or in vitro. The problem is the B lymphocyte, they do not survive for long period of time outside the body. They die, they are natural death. So that is why hybridoma technology is used. So what is done? The specific proliferated B cells are hybridized with the myeloma cells. Myeloma cells, that means some cancerous B lymphocyte. When they are fused together, they produce a hybrid cell. These cells, they cannot survive for long, but can produce antibody. They cannot survive, they cannot survive, but these can survive. Myeloma cells can survive. They are immortal. Cancerous cells, as you know, they are immortal if they get sufficient nutrients. And these B cells, they can produce antibody, and this cell cannot produce antibody. Now, this hybrid cell will survive and can produce antibody. So, this is the thing what happens in the production of monoclonal antibody, and how to do that. This was first performed by George Kohler and Cesar Milstein in 1975. They successfully hybridized these two type of cells, and for that they won Nobel Prize in the year 1984. What they do? For understanding that, first we must understand de novo synthesis of DNA. So, what happens? De novo synthesis from dihydrofolate, tetrahydrofolate is formed, hydrofolate is formed, which combined with some precursor to produce nucleotides. Which ultimately form DNA. However, a drug called aminopterin can block this pathway, that is conversion of dihydrofolate to tetrahydro, thereby prevent DNA synthesis. Now, an alternate pathway is also there in human or mammalian somatic cells. So, that is called salvage pathway. Salvage pathway. This is called de novo synthesis. De novo synthesis. The thing already I have discussed, this is de novo synthesis. For mammalian cell, alternate pathway is there. That is, an enzyme is there that is called HGPRT, hypoxanthine, guanine, phosphorybosyl transferase. This enzyme, they can utilize hypoxanthine to produce nucleotides. Purine and pyrimide is nucleotides. Hypoxanthine, this HGPRT can utilize. Another enzyme is there, TK or thymidine kinase. This can convert thymidine to nucleotides, okay, by this enzyme. So, now briefly to understand, any cell which has these enzymes can survive in presence of aminopterin, which is an anti-metabolites. So, this is the science behind the selection of hybrid cells. How? If we cultivate a cell which does not have HGPRT, will die. But if we cultivate a cell which has HGPRT, then that will survive. So, now going back to the original process. What happens? Mouse is immunized with the specific antigen. Mouse is immunized with specific antigen. Antigen is injected to the mouse. So, this will produce B-lymphocyte. B-lymphocytes. Many B-lymphocytes. Not only the specific B-lymphocyte, other lymphocytes will, that is the antigen which you are injecting, against all its epitope, the B-lymphocytes will be produced. These B-lymphocytes will proliferate to produce antibodies. Now, these B-lymphocytes we are not mentioning which B-lymphocytes, all the B-lymphocytes against all the epitopes of this antigen. So, effort should be on so that our desired B-lymphocyte is produced in higher quantity. Along with this, we take some myeloma cells. We take some myeloma cells indicated B-lymphocyte as B, myeloma cells. These are, as I told you, this cancerous cell, and they are HGPRT minus, that is, these cells does not have that enzyme HGPRT. However, this is the somatic cell, human cell, so it has HGPRT plus, that is, they have that enzyme, and this can produce antibody, and this cannot produce antibody. So, now B cells and myelin suspension are mixed together in presence of PEG, polyethylene glycol. PEG means polyethylene glycol, or initially inactivated Sendai viruses also used. Okay. So, now in presence of PEG, these cells will fuse to produce a hybrid cell. Produce a hybrid cell. These hybrid cells, their genetic material will hybridize also and will gain some properties from each of the cells. So, now these hybrid cells are HGPRT plus and can produce antibody and can survive outside our body. Three properties. This. So, now our suspension mixture, our suspension, three types of cells are there, because all cells will not hybridize. So, there will be B cell, there will be hybrid cells, and there will be myeloma cells. B cells originally we have taken, M cells we have originally taken. Some of them will remain as original and some will hybridize to form hybrid cells. Now, these cells has to be isolated. That is the selection of hybridoma. How to do that? We will cultivate them in a HAT media. H means Hypoxanthine, aminopterin, thymidine. Along with other ingredients, this media contains Hypoxanthine, aminopterin and thymidine. So, in this media, what will happen? What will happen to different types of cells? H cell, M cells and B cells. B cells will die their natural death, because as you know, B cell cannot survive outside body for long. So they will die their natural death. M cell will die because they are HGPRT minus. M cell will die because they are HGPRT minus, that is, they do not have that enzyme. So they cannot utilize hypoxanthine or thymidine to produce DNA. So it will die due to lack of enzyme. But hybrid cells will survive within 7 to 10 days. Only hybrid cells will survive. Now all the cells are taken and diluted, that is, all the surviving cells are taken and diluted, and they are put into microculture plate. Microculture, so this is the suspension in which lot of cells are there. Okay, suppose there are 100 cells, 100 surviving cells. So we have to dilute it to 100 ml, and each well there 1 ml should be given, assuming that each ml will have 1 cell. Or however, some well may have 2 cells or some may not have any cells. So it should be diluted such a way that 1 cell is present in each microculture well. So now these cell will gradually grow and secrete antibody. Secrete antibodies, and that should be tested periodically. Testing all the well, sample from each well, microliter quantity of sample from each well, and should be tested by the techniques like ELISA or RIA. They should be tested. Tested for what? Tested for presence of desired antibody. Suppose this well found to produce desired antibodies. So now these cells are collected and cultivated. Both in vivo and in vitro. These cells, suspension is taken and it is administered into the transgenic animal, peritoneum of the transgenic animal. So there these cells will proliferate and produce numerous cells of this specific B lymphocyte, which is capable of producing particular antibody. In vitro, they can be cultivated using suitable tissue culture technique. We can cultivate them in soft agar media or in liquid culture media. They can be cultivated, and these cells are capable of producing antibodies in the laboratory scale or industrial scale. So now these antibodies, as I told you, they are very much specific to each other, specific to their antigen. These antibodies, as I told you, are very much specific to its antigen. Antigen, antigenic site. It can recognize a small sequence of 5 amino acids to 20 amino acids. When you consider the structure of a protein, it is a huge molecule. 5 to 10 amino acid sequences is a negligible number of amino acids. It can recognize that too. So what is the utility? We can use them to target specific tissues like lung, kidney, brain, Even cancerous tissues can be targeted. So, the antibody is there, antibody, the drug you can attach to it and administer into the body, so that it can go and attach itself to the specific organ only, because this is very much specific, as I told you. It is sometimes used for the treatment of some specific diseases. It is used for the identification of infectious microorganism. It is also used for the identification of different chemicals, protein means chemicals means protein products. It can be used for specific identification. So, this is monoclonal antibody, and the technology by which it is done is called hybridoma technology. So, this is all about monoclonal antibody and hybridoma technology. Thanks for watching.